we discussed last time before we begin now the remaining part of that lecture? So anybody in the class, do you?
Can we talk about like this method of reporter for distinguishing between a species of bacterial?
Can you repeat the question, please?
A method of reporters.
Method of reporters.
What is a reporter and etc.
What is a reporter gene, you mean?
Yeah, ma'am.
Okay. See, I'm going to come to that when we talk about biosensors. Okay, because the entire concept of a reporter gene, etc., is going to come when we come to biosensors. So before that, let's finish the topic of ecotoxicology, and you will remember that we had kind of finished. I had come up to the dose response curve, and I'm just going to project this on a full slide mode. Everyone can see this. Can you
Yes,
see the
ma'am. We
slide?
can. Yes,
Okay,
ma'am.
so we will take it up from here. And so in the previous, in this particular slide, I was just trying to show you as to what the nomenclature is. Whenever you plot a dose response curve, number one, what does it really tell you? And number two, what is the importance of the LC50 value? Because like you see here, LC50 value or LD is exactly the same. D stands for the dosage, which could be a chemical per kilogram body weight in case you are dealing with rats and other things, or it could be the lethal concentration, which is expressed in terms of milligram per liter of that particular compound. So in the test, what you are looking at is that any concentration which gives you a 50% response. Okay, and this response could be growth inhibition, or it could be death in the case of, let's say, rat or something. That is called as a response. Okay, so the concentration or the dose at which 50% of the response is registered is called as either LD50 or LC50. Now, I'm just trying to show you a data which has been obtained for mortality of Daphnia. So there is a picture of Daphnia here. It's extremely important. It's a small insect, and it essentially feeds on the primary producers like the microalgae, bacteria, etc. Because when you go back to the trophic structure of an ecosystem, we said that the primary producers which produce the food are the microalgae and the photosynthetic bacteria. Then comes the primary consumer. So this particular insect, which is called as the Daphnia, you see the name is here Daphnia magna, is a primary consumer. Okay, so in many of these ecotoxicology studies, you will see that if you want to determine the toxicity of a particular compound, you are going to select representative organisms from each trophic level. Okay, that's very important. So this Daphnia is actually the primary consumer here. So if we examine the dose response curve for a particular chemical, which is ephedrine here, I will come back to tell you what exactly and where is ephedrine found. Okay, so you see as the concentration of this particular ephedrine increases, this concentration is reported in terms of log to the base 10 value, and this is the response. So you start out with a certain number of these insects. They can be seen under a microscope very clearly. So if you start out with 20 or 50 such insects, once you expose them to these different concentrations, incubated for a period of, like the time is given here, either for 24 hours or after 48 hours, then you look at how many of them have died. Okay, so for example, in this particular case, you have a certain concentration, but then the percent mortality is 0. Even at this level, percent mortality is 0. And it is here that you start to see the response. At this concentration, for example, you have 20%. And what you see in red here is the duplicate sample. Okay, so these are actually 48-hour values you will see here. And the circle represents replicate 1. And this represents the duplicate sample. So you see this is the response, and this circle looks a little bit darker here. That means both these values are overlapping on each other. The response in the duplicate samples is exactly identical. Okay, so both these, the red and this rhomboid, they fall on each other. Okay, and the data which you see in the blue Again, it comes for the 24-hour sample which you are trying to assess. And the circle and this rhomboid, they actually represent the duplicate samples. So is that clear? So this is how it gets translated into a real life experiment. Is that clear?
Yes, ma'am. Can you describe like what is replicate and duplicate here?
Oh, that means, you have two flasks in which you are going to conduct your experiment. All your readings are going to be based on the data which you are collecting from two different flasks. That is a duplicate. So in one flask, you are going to add 50 of these insects. In the other flask, again, you will add 50 of these insects. And then you are going to add different concentrations of the compound which you are going to test for toxicity. Then you are going to remove a sample after 24 hours, after 48 hours, so on and so forth. So in other words, the experiment has been carried out in duplicate at two different places.
Okay,
Is
ma'am.
that
Okay.
is that clear? Because you can't rely. Of course, it's expensive. Normally, you may want to carry it out in triplicate, at least 3 samples. But since many of these are expensive, you should at least do these tests in duplicate.
Yes, ma'am.
So now, in this particular slide, what is shown here is what is called as biotoxicity. Okay, so you see many times, like you saw in the last one, there is only insects and the mortality of which has been measured, which was Daphnia, which is an insect. Now there are several tests which are being used today in order to determine how toxic a particular compound is. And these are all called as biotoxicity because these are carried out on biological systems. Okay, and some of these which are very commonly used in the laboratory are called as the Ames test, and this was developed by the scientist Bruce Ames. It's also called commonly as a mutagenicity test. That is, if a given compound which you are dealing with is mutagenic. By mutagenic, what we mean is that it causes a change in the DNA basis. An adenine can be changed to a cytosine, or a thymine can be changed into a guanine, so on and so forth. So if there is a mutation in a base so that it changes from one base to another, that is called as mutagenic. Since many of these single nucleotide changes can also be associated with a complete non-functionality of the enzyme, or one amino acid could be changed by another amino acid, even if there is a single base which is changed. If one amino acid is changed by another one, sometimes it can affect badly the properties of a very critical enzyme, and it can result in cell death. Okay, but such a change which occurs at the DNA level, okay, is called as the mutagenicity test, and I will explain as to what is the basis for that. Then there are tests which are going to change, which are going to test the carcinogenicity. Okay, there is a difference between the term mutagenic and carcinogenic. Carcinogenic means it is cancer causing. That means you could have some mammalian cells in a tube, and suddenly if you add a chemical, there is such a drastic change in the DNA composition that these cells, they become cancerous. In other words, they start to multiply much faster. Now, all mutagens, they do not lead to cancer formation. There could be change at the DNA level, but it's not necessary that the cell is going to become cancerous. Whereas when you check for the carcinogenicity test, you are actually checking the potential of that contaminant to cause cancer and to have confer cell proliferation activity. So these mammalian cells in the culture plate which you are testing, they could start to multiply at a much faster rate. That is carcinogenicity. The third test is the Daphnia test system, which I already told you about. Then fish toxicity also, because eventually, many of these contaminants which are released into the water body, they enter into the food chain. They can be engulfed by fish also if they prey on small fish, if they prey on these primary consumers, and the contaminant also reaches the fish. And fish are going to be consumed by human beings. Okay, so that way it can enter into the human system. So therefore, in order to determine that, you perform what is called as the fish toxicity test. That does this contaminant, really enter into the food chain? Because then the chances are that it must have reached human beings also.
Ma'am.
Yeah, please go on.
Ma'am, in the carcinogenicity test, ma'am, the cells are multiplied in the in the rat.
Well, they can be carried out. So you don't have to go to model organism. Yeah, sometimes you do that. You may actually inject that particular toxicant into a rat and observe after a period of 20 days or 30 days whether there is a tumor formation. But generally what they do these days is that they want to minimize the use of experimental big animals like rats, you know. So they do mammalian cells which can be cultivated in an agar plate. So mammalian cells can be cultivated in an agar plate. Generally, the doubling time is about 24 hours for the normal cells. Okay, and you can also take this agar plate under a microscope and you can count the number of cells. Okay, the eukaryotic cells are very, very well defined mammalian cells. So if you see that the number of cells which you get in 24 hours is much more than what is expected, then you can conclude that that particular contaminant is a cancer causing agent.
Yes, ma'am. So, ma'am, can that contaminant be passed on in the food chain? Let's assume that if humans are consuming these fish, and can it have that risk to pass
No,
on in the humans as well?
no, not necessarily, because it is not necessary that, the somatic cells are very different and the reproductive cells are very, very different. Remember, all the reproductive cells, they are haploid. First of all, they have all become haploid. They have a single copy of the chromosome. Whereas all your somatic cells in the other body, the intestinal cells, the heart cells, or whatever, they are all diploid. So those cells are very, very different. And it's not necessary that what you put in the intestine, what gets circulated, it may reach the gonads also. It can affect the reproductive cell line also. That is a possibility. But
Yes, ma'am.
offhand,
Understood.
understood? Okay. So now let's look at this
So,
AIMS
ma'am,
test.
in carcinogenic test, they are testing that whether there is unexpected growth or not, right?
Exactly. Because cancer means rapid proliferation of cells.
Right, right, right.
Okay. So now let me first discuss the AIMS test with you. Have you read? Because in microbiology, you are expected to have read. Can you recall, remember how is AIMS test carried out?
Ma'am, we have not read, I guess.
Okay, so very quickly, in the AIMS test, what is utilized is a bacterium which is called as Salmonella typhimurium strain TA98 and TA110. Okay, now these Salmonella typhimurium strains that you get, for example, from a culture collection when you carry out AIMS test, these are all histidine negative. Like it says here, that means these particular cells are not capable of making histidine. They are defective in one of the enzymes, which is a part of the histidine biosynthetic pathway. Okay, so therefore, when you plate out, this is a cell suspension of Salmonella. When you plate it out onto a medium, you must supply histidine from outside. Otherwise, the cells will not be able to grow on minimal medium. Okay, these cells, they can grow on a medium like LB, Luria broth, which you normally use because Luria broth supplies all the amino acids. But if you grow it on minimal medium and you do not supply histidine from outside, then this Salmonella typhimurium His negative strain will not be able to grow on this petri dish. You see right here. So this is a culture plate with the growth medium, but the growth medium lacks histidine. So when you put this on top of it, you don't see any cells which are growing. Okay. Now, next what you do is that I will tell you what is the significance of this. Next, the Salmonella typhimurium into this cell suspension, you are now going to add a chemical and you want to check its mutagenicity. So you add 100 microgram per ml of a compound A and you want to check if it is mutagenic or not. Okay, so you mix it with this cell suspension, again you pour it out, or as is explained in this particular slide, that you place the chemical to be tested in the well. Okay, so here is, you have already plated out these cells. You see they are plated out, but there is no growth because the original strain is His negative. and you didn't add histidine from outside. And now you make a little well in this agar plate and you put the chemical, you have an aqueous solution, you could have an alcohol. If it is a hydrocarbon compound, you will solubilize it in an organic solvent, small amount, and you just put it in a well. And you allow this by diffusion to diffuse across the plate. Okay, and suddenly you see after incubation for 24 hours that there are some cells which have started to grow. Okay, and this is the control. That is, this is a control in which you do not add this particular chemical at all, and you just incubate it nevertheless, and you see that there is okay. There is only for a compound which is not mutagenic. You could have just one particular colony, but in this one you have a lot of these colonies, and the colonies will grow. Why? Because there are mutations which have occurred because of this chemical, as a result of which the gene and the gene which is coding for that particular defective enzyme, which is a part of histidine biosynthetic pathway, also mutated back. And that gene, let us say it was gene A in the histidine biosynthetic pathway, is reverted back to a normal protein, as a result of which it no longer requires histidine. So therefore, it is able to grow on histidine minus plate. Is that logic clear to everybody?
Ma'am, just one doubt. Ma'am, the culture plate has with growth medium, as the slide says, it lacks histidine. So, ma'am,
Exactly.
in the
Yeah.
second part, when it was written that place the chemical to be tested. So, where does from where the histidine comes from?
No, you see, the cells themselves, which were histidine negative, now have become histidine plus.
Okay.
What it means is that earlier, okay, when you say a strain is his negative, that means it is lacking in one of the enzymes, which is a part of the histidine biosynthetic pathway. Let's say histidine starts from one of the intermediates of the TCA cycle. Pick up anything, alpha ketogluteric acid. Okay, so there are three enzymes which will convert alpha ketoglutarate into intermediate 1, intermediate 2, intermediate 3, and then to histidine. That is the way the cells synthesize all of these amino acids, starting with glucose. Okay, so it is lacking in protein A. But now, by lacking, what we mean is that the gene is there, but it is not functioning. You have not deleted the gene. It is just that the gene was defective. But now, because the chemical is acting on that particular enzyme, you have exposed your cells to a certain chemical which entered, it is mutagenic. So therefore, it causes mutations in a lot of genes. But what you are focusing on is, as a representative, you are using the histidine A gene. So the histidine A also reverts back. Many mutation events are occurring in the cell, but what you are choosing for are those cells which are now capable of growing on minimal medium which lacks histidine. Okay, so these cells which you see now suddenly growing after they have been exposed to this chemical, that is because this chemical was causing mutation in the cells, and in all these handful of cells, in these 20 colonies which you are seeing, out of 1,000,000 cells which you have plated out. Okay, these 20, in this particular mutagen caused a mutation in protein A, so that now the functionality of the gene was restored back and it could synthesize now histidine from alpha ketoglutarate. Is that clear? So we
Ma'am,
see.
clear, but the thing is that how it can't happen all the time. It may mutate, but like histidine plus will histidine gene may not be restored always.
Absolutely,
It's
no,
very
but
less probability that histidine will be that the cells will be histidine positive by mutation.
Yeah,
They're
you see, because yeah, the system has been checked. I mean, everybody, even in our laboratory, we use Ames test normally. Okay, so these are very reliable strains, and these are all point mutations. They contain histidine A mutation, but these are all point mutations. So there is a single base change, which is very stable. All right, and when you plate out the cells, you are plating out nearly 10 to the 8 or 10 to the 10 cells on the culture plate. And what you are looking for on this particular plate, okay, is now something which will be able to grow on. Since you know the original strain was only negative for histidine, you are looking for unique mutational events, and out of which you get only 20 colonies which are able to grow out of maybe 10 to the 8 cells which you had plated out. Look at the probability. But since you have a very strong selection, you are able to pick up that unique event which has occurred in the histidine A gene. And that is good enough for you to know that whether the chemical is mutagenic or not.
Yes, ma'am. Understood. Understood.
Okay, so you say, you look, 20 colonies have grown here, and you may have plated 10 to the 8 cells, 10 to the 10 cells. But then it doesn't matter because so many bacteria, mutations are constantly occurring. Now, what is here is like, a spontaneous event. If you put water, for example, here, then also some of the cells are going to revert back. So the event which you see here is, if you put not the chemical in a plate, you're going to put control sample. Let's say, you have made your, this particular hydrocarbon is solubilized in carbon tetrachloride as an organic solvent. There has to be a control. So in the parallel control plate, you have only carbon tetrachloride. In this, you had a carbon tetrachloride in which your aromatic hydrocarbon was suspended. So here, you are getting one colony, but here you are getting 20 different colonies. So the higher the number of colonies, that means the compound which you have here now is inducing more mutations. That is why you are getting the number of revertants to be higher.
Yes, ma'am.
Okay, so that is what the Ames test is, and it is very routinely done in the laboratory. Like in my lab, we work with the textile effluents. So if you carry, if you just have a sample of these effluents which are generated from the textile industry and you perform Ames test, you see hundreds of thousands of colonies will appear because the textile effluents, they have azo dyes, they have so many of these other dyes which are highly mutagenic. Okay, so now when you say carcinogenicity exact, now, of course, carcinogenicity, you will not use salmonella, but you will use human cell lines, or you will induce that compound directly into rat. Now, you see, there is a variation of this test which is presented in this picture, where you have salmonella, which is mixed with rat liver extract. Okay, can anybody take a guess as to why we should have this extension of the test in which rat liver extract is also added? Can anybody take a guess?
It is added as a medium to grow for growing this salmonella.
No, not at all. Not salmonella can grow by itself. Now you see many of these toxic compounds, okay, they may not be mutagenic, but then human liver or a mammalian liver can release certain enzymes which can convert these toxin A compound into something which is more toxic. Do you understand? So what you can do in this extension is that the salmonella, it is mixed with rat liver extract in this medium. Okay, then you want to check, because what happens, you are eventually trying to test whether it is going to be carcinogenic or it's going to be mutagenic in the human system. So you have to replicate that human physiology in the test. And that is done by taking, obviously human extracts cannot be taken, but rat is a mammal. So the liver extract of the rat is taken and it is also put in the medium, so that if, for example, this particular toxicant by itself may not be mutagenic. However, in the presence of the rat liver extract, this compound A could get changed into compound B, which could be more, which could be actually mutagenic. So you also check that you have an extension of that method as to how human system is going to respond to that particular toxic compound. So is that clear as to why there's an extension of this salmonella test by incorporating rat liver extract?
Oh, yes, ma'am.
Okay. So now, let me move to, so all the tests which we have seen in the previous slide, the aquatic fish toxicity, or using the rats, etc. First of all, they are very expensive, and then you need to maintain a stock of rat, you need to maintain a stock of fish in order to carry out. So across the laboratories, globally, there was a consortium, there was a group of scientists who came together, and they said, let us try to standardize all of these tests so that test kits can be provided by a commercial manufacturer, which are going to be what is called as culture independent. That is, you can simply take that test, you can open a vial, and you can add your contaminant, the toxicity of which you want to check, then you can report your results so that there is no variability from one laboratory to another. So there was a need which was felt to carry out this test, because depending on when you are cultivating Daphnia, when you have fish, you need to maintain very large cultures. Like, for example, if I want to do aquatic fish toxicity test, I cannot carry it out in my lab. I have to go to some other lab. I have to outsource my work where fish stock are kept or where rats are kept, so on and so forth. Okay, but what if I want to do it in my own laboratory under very standard conditions? So there was a need felt to develop what are called as the standard microbiotests, which are called as culture independent. That means you don't need to have a bacterial culture and then you need to plate it out on LB, so on and so forth. So these microbiotests, and I'm going to describe these to you in the next slides. So they were actually developed by a consortium of scientists for routine monitoring of the toxicity of chemicals which people are dealing with. And the testing was decided to be done on the undiluted samples of natural waters. So if there is a natural water body, okay, which is like a lake or which is a pond, then you will just take a sample. You are not going to dilute out that, and then you are going to measure the response of a particular, whether that lake is going to be, is it contaminated with chemicals, or can it be said that it is very clean based on the tests which you are going to perform in the laboratory. Okay, or if you are dealing with wastewaters, then, like for example, there are in Yamuna, there are a lot of these industries which are discharging their water. So those wastewaters, they need to be diluted. So the standard tests are performed in a way on the wastewaters so that you determine what is called as an LC50 value. Okay, how much of dilution of that wastewater has to be carried out in order to get elucidate, or in order to have a 50% response of a test organism in terms of survival. So I will explain it to you when I come to describe the details of the test. And based on the tests which you are going to conduct on these standard tests, there is a scoring system which is going to use 5 classes. Okay, so there are water bodies which are either the natural water bodies, or these are water bodies where wastes are discharged. For example, they can be classified into 5 classes as you see in this table. Okay, these are the symbols which are usually written next to that in order to guide people. So when you say it is a class 1 lake, that means there is no acute hazard. And what does it translate to? That means that in the standard tests which you have performed, there is less than 20% inhibition which has been detected on any one of the standard tests you have performed. Okay, so remember the dose response curve. If the response is less than 20% by any one of, so if you conduct 4 different standard tests, in all the 4 standard tests, the response is less than 20%, then you say that this particular lake or a water body can be classified as a class 1 lake. That is, there is no acute hazard, and the symbol is going to be this. That means it is also fit for swimming. It's also fit for drinking water. So is that clear to everybody?
Ma'am, what is PE stands for?
PE is percentage effect. Okay,
Okay,
so when you say 20%, that means that particular toxin that you are going to test is going to evoke a 20% response on the model organism which you are trying to test it against. Let's say you have the Daphnia test which you are performing. So if you add that particular chemical to the Daphnia test, the mortality which you get of Daphnia is going to be less than 20%. If that is the case, then you say that the water body can be classified as a class 1. However, the test is not performed only on Daphnia, which is actually a primary consumer. It is going to be it could be on fish also, which is a secondary consumer. It could be on microalgae test also, which is actually your primary producers. So at different trophic levels, the tests which you have performed are going to evoke less than 20% response. Then that water body can be classified as a class 1 water body.
Ma'am, so the test is being done on undiluted samples.
Yeah, natural water body. Okay, but if it is from the wastewater, I will explain to you in the next slides. I will give you all the details of test. In the wastewaters, you are supposed to make a you pick up 20 ml of sample from Yamuna, from a part of Yamuna where a lot of industries are discharging. Okay, now when you see the original undiluted sample of that wastewater, you are going to have 100% kill because it is going to be toxic. So what you are going to do, you will make dilutions of that water body. You will make 1 to 10, 1 to 20, 1 to 50, 1 to 100 dilution, and you will see what is the dilution at 50% response. And that dilution is taken to determine a value which is called as the toxicity unit, or TU. So in the wastewater sample, what we are going to be looking at, the response will not be PE, but the PE numbers are translated into TU numbers, toxicity units. Okay, let me move ahead, and then when we come to that slide, you'll be able to understand. But as for now, so is this clear as to what is the logic for classifying them into these 5 different categories? So less than 20% response, either by microalgae or by Daphnia or by fish, will classify that as a class 1. No acute hazard. However, if the response is between 20% and 50% in any one of the tests, okay, then you will say that it's a class 2 water body. If the response, if the percentage effect is between 50% and 100% for any one of the tests, then you say it's a class 3 acute hazard, and you are going to put this kind of a symbol. Okay, and if the percentage effect is 100% in at least one test, because we are going to perform several tests. You will have test on microalgae, you will have test on Daphnia, you will have test on the fish. From all levels, all trophic levels, you are going to carry out. If the response is 100% in even one of those tests, then you say class 4. But if 100% response in all tests, then it is classified as class 5. It is called as very high acute hazard, and it is labeled by this. So this is a standard way of classifying. So is this clear to everybody? Can we move ahead?
Yes, ma'am.
Yes, ma'am, we can move ahead.
Okay. All right. So now in these microbiome tests, which are commercially available, they are sold by a company which is patented by, the group of people who actually standardize all of these tests. So what all do the tests have? First of all, they contain freeze-dried organisms or cryptic life forms. So if I were to order this test from this company, okay, I don't have to cultivate them, because in a while you are going to have freeze drying is a process whereby these can be desiccated under very low conditions, and still the cells are able to survive. Like bacterial cultures, you are storing in glycerol at -20, or you have many of these yeast and other things which you can store at -80 degrees, and still these microbes are able to survive. When you take them out of a -80-degree freezer, you thaw them, and then when you put it on a nutritional medium like YPD or LB, they are able to grow. Okay, those cultures are called as the freeze-dried organisms. So the tests which are performed are, first of all, a 72-hour algal growth inhibition with defined algae, which are written here. This is called as an algal tox kit F. Okay, but in this test kit, what is supplied are the freeze-dried algae. You will just take take them out of the vial, you will thaw them, and then you are just going to put it in a test tube, and you will add your contaminant which you want to check the toxicity for, incubate it for 72 hours, and then these are all microscopic algae. So OD you will measure at 600 nanometer, which will be a reflection of what is the increase in the growth. If the OD stays the same as it was at time 0, that means there is no growth, but that is, there is a growth inhibition. And in the control sample where you are going to add water or something like that, you will certainly see that algae is going to proliferate and you will record a certain number with respect to the control. Okay, so such a toxicology kit is called as the algal tox kit. Similarly, the second test which is normally performed is a 24-hour ciliate, which is like a protozoa. Okay, the growth inhibition, the model organism which is chosen is Tetrahymena thermophila, and this kit is called as the Protox kit F. Then rotifers, which are also very, very important components of the food chain. I will explain it to you. So like Daphnia, or like not Daphnia, but these rotifers are a different category of insects, and these also reside at the level of primary consumer level. Okay, so this is called as the Rotox kit. You are going to check 24-hour mortality, 24-hour crustacean mortality with the Daphnia, etc. The picture of that insect I had already projected to you, and you will see whether they die within a 24-hour to 48-hour exposure to the chemical or to the water body which you are testing, and that is called as the DAFTOX kit. So let me give you an example here of the first case. So if there is anything which you want me to stop at and explain, I can do that. Okay, so should we talk about the algal tox kit?
Yes, ma'am.
Okay, so here is a microscopic picture of this algae. So these are the freeze-dried cells. You see, these are all unicellular. Okay, and this, the algal tox kit F, it makes use of unique disposable long cell exposure chambers, which also serve as measuring cuvettes for direct determination of algal growth by OD measurements. So all that you have to do is, for example, this can be put, there is a vial which you just empty out, and 10 centimeter path length cuvette, you just put them out and you add a certain amount of the water, of the sample of the water which you want to test. Okay, incubate it at a certain temperature, and then after 72 hours, you are going to look at the algal growth by monitoring optical density at that particular nanometer. Okay, so in a control culture where you are going to add just simply water or some nutrients as they are specified in the kit, you will expect to see a certain growth. But then if you add, for example, your water body, you will see what is the difference in the toxicity. Okay, and you see that if you just read, for example, in this particular paragraph, that the test organisms, that is this particular microalgae, is included in the kit as algal beads. So this is the way the samples are taken. They are immobilized in these beads, and this way they can be freeze dried and they can be supplied, and the shelf life is very, very long. So I can simply order these, and then once the algal beads, for example, they are very easy. You can the microalgae can easily be set free. So if you just dissolve them in certain compounds, the algae are released from the beads, are going to dissolve. Okay, and then the algae are going to be released. So this is how the algal cells are going to be looking like. Okay, and that's the way the test is going to be performed. Okay, so when you go back, and when you say, what am I really doing in this microbiome test? I'm going to do a 72-hour algal growth experiment with Selenestrum, or this one, or some other algae which has been used. So, so here is a picture of that second organism which is listed on that slide. This is the way that they look like under microscope. Just as you do for bacteria, the eukaryotic cultures like microalgae can also be, the growth can be estimated by monitoring optical density. So I hope it is clear to everybody. Now quickly, let's quickly move to, so the Protox kit F. Okay, here is a picture of the paramecium. And in the same way, the paramecium, for example, is tested. But here the principle, just read it, the principle is a little bit different, because here what you do is that in order for paramecium to grow, a food solution is supplied in the vial. Okay, so that food solution has a
Ma'am,
certain OD. Yeah.
you talked about like tetrahymena. So like this is tetrahymena or
Yeah, yeah, yeah.
paramecium. Like
No, no, no. This is a tetrahymena,
okay,
Thermophila,
ma'am.
but it is a protozoa.
Okay, okay, okay.
Okay. And what is the importance of these protozoa in the ecological chain? You will recall that they are the primary grazers. They feed on microalgae and they feed on photosynthetic bacteria, etc. So they make a very important connection between the primary producer and the secondary consumers. Through trophic chain, how does the energy really move from primary producer to a secondary consumer? It is this primary consumer which transfers the energy from microalgae, from photosynthetic bacteria into the second trophic level, like the small fish. Okay.
Yes, ma'am.
Exactly. So that's how we do. And like I said, that in this particular case, what you are doing is that these are going to be suspended in some food solution, and there is a certain OD, which is measured, you measure the OD at 440 nanometer, etc. And then what you do is that after some time, as the protozoa multiply on the food, the overall density will go down. The overall OD will go down because the turbidity of the solution goes down because food is being consumed. So a decrease in the optical density. Here you are not enumerating these particular microbes. Okay, but rather what you are doing is that you are measuring a decrease in the optical density at 440 nanometer, which is an indication of the fact that the protozoa are growing and they are consuming the food. That is why the OD440 value is going down. So is that clear? So that is how, for example,
Yes, ma'am.
these tests are carried out. And then you have what is called as rotokit. This is another important, this is a primary consumer, like you see here. And just fascinating that, this test organism is included in the kit as cysts, which are actually dormant eggs, which can be easily hatched on demand in less than 20 hours to supply the live organism to conduct the toxicity test. So again, when you order for the kit, like you see here, for example, the Roto X kit, you see here, all these vials are there. And then all you have, they contain the dormant eggs. So you just add something to it and all of these microorganisms will be released and you will be able to carry out. So you don't have to do any culturing inside your laboratory. Okay. Now, again, the importance of rotifers in the, why do we choose? The question is, you may wonder, why am I choosing these? What is the logic behind it? The logic is that they are highly representative of a trophic level. So rotifers are important in freshwater environments. They have the highest reproductive rate among the metazoans, which is a category of animals at that trophic level, thus obtaining very high population density in a short time, being dominant in many zoo taxonomy. So you don't want to be dealing with an animal in the laboratory where it takes them, many hours to go from a number from 2 to 4. Okay, so here, they can multiply very rapidly in a short period of time. So in 24 hours, they may have gone many doublings. So you will be able to see the effect of that particular contaminant on this body much faster. If it is going from 1 to 2 or 2 to 4 and you suddenly see 3, you are not able to make a conclusion. But if it is going from 2 to 24 in one case and in the other case it is only 2 to 4, you are able to make a distinction very clearly about the toxicity of a compound. Okay,
Ma'am,
so
I have a
this,
question.
yeah.
Ma'am, like this is basic question. Like, what is metazoans? Ma'am, I
Don't
don't
worry.
know.
Do not worry, you're not supposed to know. It is only for a scientific description, because I mean, metazoans and protozoa, etc., etc. These are certain categories which are defined in zoology, and it's a highly specific scientific.
No, ma'am. The thing is that like
All
I
of you
did
is interested, I will
it came
explain.
in other course also. Like I was reading, then I searched for this. Then I got like animals are described as metazoans. Then another
Yeah,
at another like in bioinformatics class, I was like searching on. Then in metazoans, like only small organisms like were there, and other like
I
higher
see.
organisms were not there. So that's why
I
I
see.
was
I see.
confused.
So you mean to say it is a time period during which I'm not really, I have forgotten. I have read it, of course, long time back, but I cannot recall the exact definition. Does it refer to a time period, or is it a classification system?
It is in taxonomy, definitely it is a classification systems. Like certain kinds of organisms are considered as metazoans, but like I
Right.
don't know like what
Yeah.
are
Yeah.
what is
Don't
the class
worry.
of that.
Yeah, don't worry. I should have been able to explain it term down here, but don't worry. I think you can just ignore that. I will only say that at the primary trophic level, at the primary consumer level, it has one of the highest reproductive rate among, because metazoans must be occupying that particular trophic level. And amongst those, you have this as a model system. Okay, so you can get very high population density in short times, and certainly, it allows you to do your testing also in a short period of time. And they also act as a link between the microbial community, which is at the primary producer level, and the higher trophic levels. And they colonize habitats very quickly. They convert the primary producers, which are algae and cyanobacteria, in a usable form for secondary consumers, therefore making energy available for the next trophic level. Extremely important. Okay, because in all these tests, please pay special attention. You are choosing organisms from every trophic level. And that's the reason why in ecotoxicology we say we are checking the toxicity of the ecosystem, that is, all representatives of the ecosystem. And that's why you are not just saying a toxicology test, you're saying an ecotoxicology. And interstitial water from swampy soil, they also contribute to nutrient recycling. Okay, because these are zooplanktons, they take up nutrients, the carbon is metabolized and carbon is recycled back, because everything goes out in the form of carbon dioxide. Or they take up nitrogen containing foods and they return it in the form of ammonia. They discharge ammonia, collect all the nitrogen from proteins, from it in the form of ammonia and excrete it out. That is nothing but nitrogen cycling and carbon cycling. When we learn about cycling of these elements, you will see that all these microbes, such insects, they have a huge role to play, because they are rapidly growing, they are rapidly degrading. Okay, so they also accelerate the process of nutrient cycling. I mean, you don't want to have all the carbon dioxide of atmosphere trapped in a tree forever. You want it to be recycled back into carbon dioxide. Okay. So is that point clear until now? Do you understand what we have said so far?
Yes, ma'am.
Okay. So anybody else in the class, do you understand what is the importance of these microbiome tests and how they are, taken from each trophic level of an ecosystem?
Yes, ma'am.
Yes, ma'am, because they are connections between two different consumer levels, so they are important in the environment to energy transformation in upper levels in food
Absolutely.
cycle.
Absolutely. And when you want to talk about toxicity, you have to be worried about how it is impacting every trophic level. That is why
Yes, ma'am.
representatives from every trophic level need to be taken. So here is just like so a lot of laboratories over the world, they have now there were tests which were there earlier. And what people have done is that they compare across laboratories using the microbiome tests. So it is the data which is given here is like intra and inter-laboratory sensitivity comparison study using this DAFTOX kit F Magna. We already saw that they make use of these particular insects, which are the Daphnia. And the name of that kit is called as DAFTOX kit F. Okay, and it was used for checking the toxicity of pesticides in Poland, household products in Croatia, waste leachates in Australia, so on and so forth. Okay, so I want you to just look at this particular table where the standard lab cultures were taken. So people, the older methods were taken in this study. Then the new microbiome test, which uses Daphnia, was taken in the year 2003 and 5, and then ISO, which are the international standard tests which are there. The number of participants, 16 labs, 33 labs for the Daphnia, 56 labs, 40 labs participated, and they determined, for example, for certain particular compound was performed, like potassium dichromate was being evaluated, and DAFTOX was used. And the data was compared between the lab cultures, between the Daphnia and the ISO, which was announced earlier. So now if you look at the mean 24-hour EC50, okay, remember that this is the effective concentration. So the concentration at which you saw 50% mortality of the Daphnia was compared among the different tests. And what you found was that if you use the earlier lab cultures, which people were doing, the values were 1.08 milligram per liter. Okay, with the DAFTOX, which was performed in 2003 and 5, the values were 1.08, 1.20. Okay, and then with the ISO standard test, which is provided, the value was 1. In other words, the DAFTOX was working very, very well. And they were very inexpensive to perform, easy to perform. There was no culturing which was required, as opposed to these other tests. So these were pretty good, and the response was very good of these tests. And then the number which you see here was the coefficient of variation, which is actually the degree of precision of the assays was as good or as bad as it is for the other tests. So with the DAF text was just as good as this. Okay, so it gives you an idea that, all these tests which are now available, they have also been tested. So now, using these tests, let me just give you some case studies which have been done. So the paper which I'm going to be discussing is now a practical and a user-friendly toxicity classification system with microbiome tests for natural waters and wastewaters. Okay, so so this table, I already showed you when I talked about the microbiome tests. Okay, so this is a hazard classification system for natural waters, and I've already discussed it with you. This is exactly the same table which I explained. Okay, and what you look at PE is the percentage effect, and how the values which are determined in terms of percentage effects are now transferred into a classification system. So this is the way they are transferred. Okay, now in addition to that, in the same tests, there is what is called as a test score which is given. Okay, so if the effect is no significant toxic effect, then it is assigned a value of 0. So for example, if any toxic compound, the value is less than 20%. So for that chemical, the score is going to be 0. But if the effect is between 20 to 50%, then it is assigned a value of 1, so on and so forth. And then we look at like the class weight score is calculated, and there is a class weight score as a percentage. Okay, so please read all of this together. But now I'm just going to shift it to how these tests have been used and the data which is generated. How does it allow you to classify that natural water body? So is it okay? I think so. Please pay attention to this slide. So here is a natural water body. It is Valgejogi River in Estonia, where the water sample is removed. And remember that there is no dilution done because these are the natural water bodies in a country. Is that clear to everybody?
Yes, ma'am.
Okay, so this is one river
Yes,
sample.
ma'am.
It's a river sample. When you carry out the microalgae test, okay, you see that the percent effect is 0%. That is, when this natural water sample was taken and you performed, you measured what is the growth inhibition of this microalgae. There was no inhibition of growth. And that is what we mean by 0% effect. If that is the case, then the test score which is assigned is going to be 0. Okay, let me quickly take you back to the last slide. So if there is no significant toxic effect, it is assigned a score of 0. Now, for the same water sample, you carry out a test on vascular plants. Okay, so this is also one of the standard tests which is used. It is a conventional bioassay where a plant is taken and you see whether there is inhibition in the plant growth, etc. But the same water sample gave 23% effect, 23% inhibition of growth of this vascular plant. Okay, the test score which was assigned to this was 1. Why? Because if the effect is between 20 to 50%, then you assign a score of 1. So the test score which you are going to assign is going to be 1. Then you did tetrahymena thermophila test, which is by a single asterisk, by the tox kit, which I just discussed with you, all those 4 different tox kits. Percent effect is 3. Since it is less than 20%, the test score which is assigned is 0. When you do the Daphnia test, again, there is no effect. So there is 0 inhibition. The cells were able to grow as well as the control sample was. Test score is 0. Okay. Now, in one of the tests, the effect was 23%, which is more than 20%. So you are going to call it as a class 2 hazard. Okay. Remember, this is how we defined. If our effect in any one of the tests is more than 20% but less than 50%, then I'm going to classify that water body as a class 2 water body, which is slight acute hazard. That clear to everybody? As to now, let's examine the data for the first category for this particular river. It is classified as a class 2 because in one of the tests it showed 23%, that is slight acute hazard. Then there is another way of classifying which was given on the last slide, which is called as a class weight score. That is, whatever the weight scores are for each category, they are combined and they are averaged out. So you have like a test score of 0 plus 1 plus 0 plus 0. So total of 1 divided by 4, which is the sum of 4 test scores which are carried out. So the average class weight is now 0.25. Is that clear how you get that number?
Ma'am, can you explain again?
Okay, so then there is another way of classification which is called as a class weight score. Okay, so in this what is done is that the weight score which is assigned in each category, you have 4 tests which you have performed. Okay, in one of them the percent effect was 0, test score which is assigned is 0. Where does this number come from? Okay, because there is an allocation of a test score here. If the effect is less than 20%, then you are going to assign a score of 0. That's where this 0 comes from. If the
Yes,
effect is
ma'am.
between 20 and 50, then you assign 1. So when you do a class weight score, you are just going to add up all these test scores 0 plus 1. So please hold on for a minute. I just Okay, so can you see the slide again now?
Yes, ma'am.
Okay, so so this is how class weight score is calculated. Then there is a class weight score as a percentage. And how do you really compute it? You take 0.25 from this particular data set and divide it by the highest test score. So if you examine this, the highest test score is 1. You divide it by 1 and you get 25. In other words, one particular category, okay, is contributing to 25% of the overall score. So that is the class weight score. Now, the same thing can be done for the second sample here, which is for Crudy book. And the results are shown here. Now, you see, again, 4 tests are performed on this particular water body. Brooks are generally natural water bodies. So when you perform, you take that water sample from that water body and you have performed the microalgae test, you perform a test, this vascular plant, which is a conventional bioassay, then the protozoan test, then the Daphnia test, etc. Now, in one of the tests, okay, this water body sample, it had 54% inhibitory effect. Here it had 90%, here it had 46, and here it had 55%. Okay, now such a water body will be classified under class 3. Why class 3? Okay, because in one of the tests, you see here, go back to this table. In one of the tests, you were able to see a percentage inhibition or the percent effect was between 50 and 100%. That is why it is classified as a class 3 acute hazard. So you see, it is classified as a class 3 because in one of the 4 tests which you have conducted, the values were between 50 and 90%. So is that clear to everybody?
Yes, ma'am.
Okay, so in the same manner as you did class weight score here, we will do the class weight score for this water body also, and the test scores are drawn exactly from the same table. So if you have like less than PE 50, you give score of 1. If it is between 50 but less than 100, you give a score of 2. Okay, so based on exactly the same way, because you are between 50 and 90, you give a test score of 2. And you are still less than 100%, you give a score of 2, less than 50%, 1 and 2. Okay, so these test scores are averaged out. You see, you have 2 plus 2 plus 1 plus 2. So all of the test scores are added to 7 and they are divided by 4 number of tests performed and the value is 1.7. And then in the class weight score as a percentage, you have 1.75 which is divided by 2, which is the highest score which you get, which was actually 2. So that way you get 87.5% as the class weight score.
Ma'am, what does the class weight score represent physically, ma'am?
Okay.
Can we?
It represents that to the overall toxicity, there is one particular component which is contributing 87.5%. That's what it means. Okay.
Yes, ma'am.
Because understand, I mean, so there is at least, for example, whatever ingredient is in there. Okay, so it is contributing to the overall test score in a very big. So there could be a major pollutant in the river or something like that, which is contributing maximally to this. That's what it means. So is that clear?
Yes, ma'am, it is clear.
Okay, so now, the second part, the second one in the same paper was the hazard classification system, which is used for wastewater streams. Remember, I said that in these standard tests, either you were using the fresh waters or using the wastewaters. The reason is that in the wastewaters, the pollutant concentrations are so large that you will always see sometimes 100% inhibition, 100%. Then how do you really come to a number? How do you really classify that? Is it really very acutely toxic? Is it mildly toxic? So on and so forth. Okay, in order to do that, there is a different classification system which is adopted, and there the percentage effect is converted into a toxicity unit or TU. Number one. And number two, that if there is a 100% kill which is caused by that water body, then you are going to make several dilutions of that sample. As I said yesterday, as I said earlier, that you will make a 1 in 10, 1 in 20, 1 in 50, 1 in 100 dilution, and you will check the dilution at which you get 50% effect. And that is how you are going to calculate the toxicity. So obviously, higher the dilution you have to be making. Okay, that means the more toxic it is going to be. Because if there is a water body which is highly contaminated and you take the original sample and you get 100% kill, okay, you need to make a dilution, and the more the dilutions which you make, okay, that means the more toxic that system is going to be. And then you calculate what is the dilution at which, for example, there is 50% inhibition, and this is the way you are going to. So if there is 100%, if there is if there is a 50% dilution, 50% effect here, okay, then that translates into toxicity unit of 1. Okay, so that is how the system works. That is, that if we now look at what is the conversion in the toxic, what is the conversion in TU and the percentage effect, or the LC50 value which you are converting. With respect to LC50 value, this is how the toxicity units are going to be converted. So whatever dilution you have, whatever is the LC50 value, let's say it's 2.5 milligram or 2 or that if you don't make any dilution, for example, 100% of the sample, then the toxicity unit is going to be 2. But if you have a 50% effect, then the toxicity value is 1. Okay, so all that data is defined in this particular table, but this is for the wastewaters. So you see that if you have a 20% inhibition effect, then it translates into a toxicity unit of less than 0.4. If you have a 50% effect, then it is translated into 1 TU. And accordingly, the class, the hazard classification is carried out. If the numbers are less than 0.4 TU, then it is assigned as class 1. If the toxicity units are between 0.4 and 1 in at least one test, then it is classified as a class 2 water body, so on and so forth. Okay, so the only point which you have to remember here is that since these are wastewaters, if you carry out the percentage effect in terms of you are just monitoring 20% inhibition, 20% inhibition also is equivalent to 0.4 toxicity units. And if the value is very, very high, then you have to make dilutions of your sample, and you have to put that dilution at which you are getting 50% value, and that is translated into a toxicity unit classification. Maybe I will distribute some numericals, I will distribute to you so that you can actually see the effect between the two, or the relationship between the two.
Ma'am, the would not be counted into the toxicity unit.
Yeah, say that again.
If the water is too much jarred, in that case we have to dilute it, and suppose we are diluting it 100 times, so is that dilution factor counted into the toxicity unit?
No, not at all. But what you will do is that you will only put in this number, okay, the dilution at which you are able to get 50% kill of the test organism.
Okay, ma'am.
Okay,
But
so let's say you make 1 in 100 dilution.
Yes, ma'am.
Okay. If you make 1 in 100 dilution, that means that the toxicity unit are going to be like 100,000. If you make a 1,000 dilution of that, then the toxicity value is going to be 1,000.
Yes, ma'am.
Okay, and obviously more. But if you have to make only 1 in 10 dilution, then the toxicity values is going to be 10.
Ma'am, what
Okay.
does that line means 50% effect is 1 TU, ma'am. I could not able
Okay.
to understand.
Okay. That means 50% inhibition, because you remember that in each category you are saying 50% mortality of the test organism. So if there is a 50% mortality at that particular dilution, then in terms of the toxicity unit, that number 50% PE gets translated into 1 TU. It is equivalent to 1 TU. Is that clear?
Okay, ma'am.
Because see, sometimes till now you only looked at the PE values, but now in the case of wastewater streams, they are not using the same classification, because they are saying that the percentage, if 100%, then how do you really, you want to classify them also, that whether it is very acute, highly acute, or slightly acute, etc. It doesn't allow you to do that. So therefore you want to do a dilution of that sample. And accordingly, that dilution factor is taken into account in order to assign, but now they don't use the term PE, because that you are using for the fresh water. So they have converted that now it should be converted into a TU unit, toxicity unit. Okay, so now again, these are the examples which are being given to you from two different wastewater bodies. This is a percolate from a waste dump, wastewaters in Estonia. So two sites they have identified. And again, microalgae test is done, vascular plant, the protozoa, and the Daphnia test is given. So the toxicity unit, which is defined under the microalgae, is 43.5. The test score assigned is 3. Why? Because is test score, yeah, right here. So if you see, these are the test scores right here. So if your value is between 10 and 100, then the test score which is assigned is 3. Okay, if the value is more than 100, the test score assigned is 4. So in this case, for example, if it is 200, the toxicity units were 200, the test score which is assigned is 4. Okay, so exactly the same way. So in terms of classification, you will classify it as class 5. Why? Because in one of the tests, it has shown a value of more than 100. That's in this particular test, the toxicity unit is more than 100. So you classify it as class 5, very high acute toxicity. The class weight score is calculated the same way. So you have a 3 plus 3, 6 plus 2 plus 4, that is 12. Okay, so you have 3 plus 3, 6 plus 2, 8 plus 4, 12 divided by 4. So 3, and then class weight score is 3 divided by, excuse me. So you have an average divided by 3.
And we are not audible.
Yeah, the class weight score, if you see here, it is the average that you get divided by the highest class score, which in the first water body was 4. Okay, that's the reason why it is being divided by 4 here. The average divided by the highest class score. So 75%. So in the second example, you see the wastewaters, which are not treated anyway. So all the numbers are given. On the microalgae test, you get 1% toxicity unit. On the vascular plant, you have 6.2, so on and so forth. So the classification, the weight score, and the weight score as percentage is calculated exactly the same way as you did for this. Okay, so now I hope with this you understand that what is the application, what is the importance of this. I've only tried to summarize it in this slide, that all these microbiome tests which you have seen are very simple, and there is a rapid scoring system which you have learned in terms of either PE or the toxicity unit. It allows you to assign a score to that particular test. There are two classification systems which you have understood. One is for determination of the degree of toxic contamination of natural waters, which is called as a hazard classification. And the other one which you saw is for the quantification of the toxicity of the wastes prior to their release, and that is called as the toxicity classification system. Okay, and this entire exercise is based on microbiotests, undiluted samples of natural waters, but you have to make dilutions of the wastewaters. Okay, so please read. I will also share the original paper with you. Now I'm going to move to, like, when we talk about ecotoxicology, etc. Now, there is another category which, although I didn't get time to explain it to you, is an emerging category of pollutants. Okay, and these are generally called as the over-the-counter pharmaceuticals. Okay, by over-the-counter OTC, what we mean is that, whenever you are sick or when you have cold or something, you go to a pharmacist, you go to a chemist, and you say, well, I need Allegra, I need anti-allergen, or I need a cough syrup, for example, or I just need paracetamol, because you think that, a slight cough syrup, etc., you can handle with this. Or some people have slight asthmatic problems, so they will always keep some, some spray with them. And this is something for which a doctor's prescription is not required, and that's why it is called as over-the-counter. Is that understood, first of all? What is an OTC pharmaceutical? So hello, so is the class able to understand what is an OTC pharmaceutical?
Yes, ma'am, for which like prescription is not required.
Not required, and you can just go to the chemist and you can buy them. Now, if you look at, for example, all of these, okay, I can't recall, but let me give you an example of cetirizine hydrochloride. This is the constituent in Allegra that you take. Okay, or for example, you take this clotrimazole. It is an antifungal powder. Many people sweat during the summer months, then they go to the counter and they say, please give me a powder, okay, which can control the fungal growth. And this clotrimazole is in the powdered form, which is just used to put on, for example, on wherever you have this fungal growth, ringworms and things like that. Okay, or you have, for example, you look at this phenyl. Ephedrine is here in this category, pseudoephedrine or ephedrine, for example. So when you have you have slight cough or something, you just go to a chemist and you get a cough syrup. The cough syrup contains pseudoephedrine. Okay, so all of these are now emerging pollutants because during their manufacture, during their careless usage by people, you see that the date is already expired, the bottle is very, so you will just mindlessly, people will put it into the dustbin, or they will simply drain it out into the sink, and it enters into the water bodies. And these are the new emerging pollutants, which are nothing but pharmaceuticals, which people are buying over the counter, and they are just discharging them without any discrimination into their dustbins or into the water bodies. Okay, so many of them have become now hazardous chemical because in several studies it has been shown that they also interfere with the microorganisms in the ecological system or with different trophic level systems. Okay, so here is just a list of these. So these are, these are all listed. They are all over the counter available. And now let me just quickly go over to some of the test results which have been conducted with these. And by the way, these are all called as the emerging pollutants now. So in the chronic ecotoxicity test which have been carried out with many of these, okay, for example, you see here the Daphnia test has been performed with many of these pharmaceuticals, or aquatic fish toxicity has been carried out. On the basis of the response that they give in the Daphnia test, they can be classified as either chronic 3, chronic 1, or chronic 3 pollutant or toxic. What do these numbers mean? By chronic 1, it means that the concentration range in which they are able to show an LC50 value is less than or equal to 1 milligram per liter. Okay, so now let's take the example of like this benzalkonium chloride. So what you see is that so for this fourth chemical, you see 0.04. Okay, so that means you require 0.04 milligram per liter of this in order to cause 50% toxicity of daphnia. These are all LC50 values. Okay, if that is the case, if the concentration which is required is only less than 1 milligram, then it is called as a chronic 1. And that's the reason why this chemical is called as a chronic 1 chemical, toxin rather. Similarly, you have 15.2 here, which is a higher concentration. So you require 15, or from the data which is determined experimentally, 15.2 milligram per liter are required in order to cause 50% toxicity. Okay, if that is the case, that is if the number is more than 10, but it is less than 100, then you classify it as a chronic 3. And that is why it is classified as a chronic 3 toxin here. So is that clear to everybody? All right. So please, is that clear
Yes, ma'am.
of your classification, etc.? Okay. So now, let me stop here, and then I'm going to discuss a paper with you in which you will see how many of these, over-the-counter drugs, which are now considered as emerging pollutants, how biotechnological solutions are being found in order to address them. Because now you have a problem, and there is no quick solution to them. There is no chemical, or there is no other process, sonication or whatever, in order for removal of that. So we are now, given the advances we have now in our understanding of microbial systems, we are trying to find microbial solutions to their removal. Okay, so I will do a case study with you in the next class. So
Ma'am,
that's it. Yeah.
ma'am, I have a general query. Since minor is on 17th of Feb of BBL341. So, ma'am, what kind of questions do we expect? Ma'am, can we all the questions can be all covered with the slides since we have attended the class?
No, you will have, and you know what I will do? That last 2 years papers I'm going to share with you. I will just post them so that you have an idea of what kind of questions are likely to come.
Yes, ma'am. Thank you.
Okay, and you have in some of them the reading material has to be read. Otherwise, you will not be able to answer it completely, the question. So don't just rely on slides, but reading materials regularly I have been uploading, and please don't look for shortcuts. Go through those readings. They are not very heavy readings. Once you decide to start to work on them, just 1.5 hours per day is enough.
Yes, ma'am. Sure, I will do that.
Okay, and all of you, please. I mean, these are very, very interesting topics, and I'm sure you want to know as much as possible.
And ma'am, if we have any doubt, can I'll mail some doubt if
Certainly, absolutely.